Myself Pratiksha from 2nd MSc Applied Microbiology. My topic is sampling technique in food. Food sampling is extremely important in the food industry. Quality control is a process that helps to target, identify and rectify defects in finished food products. So this can be achieved by eliminating the source of quality problems to ensure safety. A food sample test is carried out by putting the product through a physical analysis, and this could be undertaken by manufacturer or by the official food law enforcement team, and not only for control purpose but also for research and public informations also. Sample collection technique. There are various informations are required for the sample collection technique, such as name of the food, lot number, container size, product code number, labeling information, and conditions of the lot, that is broken packages, evidence or rodents or insect infestations, etc. Quality of food samples to be collected for analysis. Different products need different approximate quality for food sampling. For example, milk required 500 ml approximately for the quality of food sample. Cheese required 200 grams. Baking powder require 100 grams. These are some of the examples for the quantity of food samples to be collected for analysis. Sample tools and container. Sample tools and containers are important for the sample collection. Sample collected from bulk packages or unpacked food sold at retail must be placed in suitable container for storage and handling to be presented for laboratory analysis. So sampling tools, which includes common tools such as spoon, screwdriver, knife, are useful for opening containers, cutting bags of food products. So specific tools are required for the specific examinations of particular food products. Next, sampling container. Sampling container, which includes for solid, deep, dry, clean, wide mouth, waterproof materials should be used. For butter, suitable wide mouth jars to be used, which should not be allowed to come in contact with the paper. So the container must be secure closure of rubber or plastic stopper, or a screw cap of metal or plastic can be used for the sampling containers. Next, source of food. Various sources of foods are needed. Bulk commodities. Bulk commodities is nothing but several samples may need to be taken from separate cases packages at several points in a container, which is similar to the random sampling, is preferable to the collection of readily accessible units. Wholesale food. Wholesale food, which includes same as the bulk commodities, and random samplization or is essential. Retail food. Retail food, which includes the food constitute the majority of foods included in food consumption database in industrialized countries. For primary food products such as meat, fruits or vegetables, the major concerns of the sampling protocol is to be ensured that the complete range of sales outlet is represented. Next, field or garden produce. These foods tends to be much more variable. The composition of food is essentially depend on the soil and fertilizer treatment. For example, for bulk commodities, meat, grain, fruit, vegetables are included, and which is also for wholesale food also. Retail food, which includes food sold to the consumer, example meat, vegetables, processed food, and which is used mainly to assess household and individual food and nutrient intake. Next, field, garden or wild foods includes food grown on gathered, hunted animals. If food as consumed, uses to the assess for individual. Next, methods of sampling. Various methods are involved in sampling. So two types are divided: random sampling and non-random sampling. Random sampling includes simple random, stratified sampling, systematic sampling and multi-stage sampling. Non-random sampling includes judgment, quota, convenience and snowball. Random sampling is also known as probability sampling, and non-random sampling is also known as non-probability sampling. First, random samplings. Random sampling is nothing but samples are taken in a way that ensure that any one unit has an equal chance of being included. This can be applicable for the nutritional database for sampling foods. Stratified sampling. Unit of sampling are taken from defined strata, which means subparts of the pattern populations. Within each stratum, the samples are taken randomly. Next, selective sampling. Selective sampling is nothing but samples are taken according to the sampling plan that excludes materials with certain characteristics or selected only those which within defined characteristics. So this is commonly used in the analysis of contaminants, and it is also used for the database work. Next, convenience sampling. Samples are taken on the basis of accessibility, expediency, cost, or other reasons and not directly concerned with sampling parameters. Sampling parameters are not considered in convenience sampling. This is usable for which is suitable for the database work, but may be the only practicable way to sample wild or uncultivated foods or composite dishes from household. So these are the most common methods of sampling. Advantage and disadvantage of sampling. Advantages, which include sampling save time. Sampling avoid repetitions of query for each and every individuals. Sampling gives nearest accurate result. So accurate results are given for the whole set of the food. And next, with small sources, we can get more data. Small quantity of amount is required for the sampling for analyzing the whole set of food products. So disadvantages include chances of biased answers. Sometimes their answers may be biased, and selection of good sample is difficult. We need to select the good sample at good. Limited knowledge may mislead the result. Sampling method may be unsuitable. Thank you.